Thank you for showing interest in our work on the effect of therapeutic ultrasound on cytoskeletal elements and cell crawling. Our experiments were planned to be done on mirroring 3LL and 4T1 cancer cells. Previous studies from our lab showed increased levels of expression of heat shock proteins at the mRNA and protein levels after focused ultrasound treatments. And these studies demonstrated that with increasing power of low intensity focused ultrasound treatment, the expression and cellular distribution of HSP gene families in 4T1 breast cancer cells was effectively increased as the amount of power of focused ultrasound treatment was increased. So this showed that focused ultrasound has the ability to modulate proteins within cells. So for our study, we looked at cell crawling, which is vital to cellular function. This challenge with cell crawling is that the machinery for cell crawling must be simultaneously self-constructing and recycling. And the adhesions at the back have to be eliminated once they become an obstacle to forward motion. So the solution to this problem is membrane tension. And Pascal's law tells us here that since the cell is essentially a fluid-filled sac, pressure exerted at the front will then be transmitted equally in all directions through the fluid, which means that the force-driven movement forward of the actin network and the rebranching of the actin network will cause force to be transmitted backwards to disintegrate the residual actin at the back, the adhesions which are actually inhibiting cell movement at that point. And branched actin is vital to cell environment exploration. Previous research has shown that chemical disruption of the actin network leads to disoriented unipolar cells that are really only able to move in one direction and can no longer integrate external signals. And other research has shown that cell migration controls the cell cycle and cell proliferation. So as cells migrate more, they actually have shorter cell cycles and increase cell proliferation. And those cells that migrate less have shorter cell cycles and have longer cell cycles and decrease cell proliferation. And what that means is that essentially, if cell crawling could be inhibited through focused ultrasound treatment, or at least reduced through focused ultrasound treatment, that could actually reduce the that could increase the lifespans of cancer cells and reduce a metastatic mechanism, as cell crawling is the primary mechanism of cancer cell metastasis. And focused ultrasound would then be hypothesized to disrupt the cancer cell cytoskeleton. And there is an essential protein pathway that allows for branched actin network sensing, because that pathway allows for regulation of increases in branched actin, which is vital to integrating signals from that branched actin for the cell. So that is the wave 1 RAC ARPIN pathway. And the first variant of our hypothesis was that the heat of focused ultrasound would denature the cytoskeletal proteins. They would then become unable to navigate, overcome obstacles, change directions, and integrate outside growth factors, which are, of course, essential to cancer cell growth. The second variant was that the heat of focused ultrasound might actually stimulate the cytoskeleton and produce motile and faster moving cells, which would, of course, be clinically relevant, because then HIFU treatment would have to be administered directly after LOFU treatment to minimize the risk of metastasis and the metastatic potential of these cells, since LOFU treatment has been shown to be essential in immunomodulation to actually help the body fight against the cancer through the immune system. So therefore, LOFU is important, but if this were true, HIFU would have to be administered directly after to avoid adverse effects to the patients. And our first question that we wanted to explore was whether focused ultrasound treatment can actually inhibit the migration of tumor cells. And we were planning to do this through a wound healing assay and a transwell migration assay with a non-treatment and a focused ultrasound treatment group. And the second question was, what are the possible underlying mechanisms involved in the inhibition of cell crawling by focused ultrasound? So we wanted to know, was it a direct denaturing effect on the cytoskeleton structure, or was it a more nuanced inhibition of pSTAT3 signaling, or mRNI-124, which is actually involved in the metastases? And to figure this out, for variant A, we wanted to do cytoskeleton staining, and for variant B, we would do immunofluorescence staining for pSTAT3. And for in vitro focused ultrasound treatment setup, we would essentially have 2 million cells suspended in sterile media, which would then be spun down in a centrifuge to form a cell pellet. This cell pellet would then be treated with the Phillips TIPS system, whereby a transducer would be applied directly to the cell pellet while it is embedded in the gel, and while the cone is filled with water for coupling. The cells would then be spun down to single cell and resuspended to make single cell suspensions and plated in 60 millimeter plates for further analysis. The cells would then be collected at specified time points, stained with specific antibodies to identify cell markers through flow cytometry. And this flow data that was acquired would then be processed to identify changes in the cell markers following our ultrasound treatments. And of course, this work was not able to be done because of the effects of the COVID-19 pandemic. But I would like to thank all my advisors on this project, Dr. Chandan Guha, Dr. Indranil Basu, Dr. Patrick Broden, Dr. Chandra Karunakaran, Dr. Sarab Singh, Ms. Claudia Gutierrez Chavez, and of course the rest of the members of Dr. Guha's team at the Department of Radiation Oncology, and also the Focused Ultrasound Foundation for awarding me the Summer 2020 Focused Ultrasound Foundation Global Internship.